we learn from Charles Darwin how, under the mechanics of natural selection, complex traits can evolve to perfection. And we can assume that most of the traits that we now observe in the living world have evolved almost to perfection. I'll show you just one example. And this already shows you that it doesn't matter from which material the process starts. So if ecology requires seeing pictures, then a camera eye is the solution. And here you see two such solutions. This is our eye, the vertebrate eye. And here you see a camera eye which has independently from our eye evolved. This is octopus eye. This is a kind of muscle. And you see the end product has the same physical perfection, even though the material from which it is developed in ontogeny is completely different. So there are hundreds of such examples that independently the perfect design has been evolved. Our predecessors have even done experiments to test Darwin's ideas. So if you go to a market, then all the fruits and vegetables that you see there have been evolved, not through natural selection, but through human selection, applying Darwin's principles. We see these different forms of cabbage are exactly those that the people had in mind who selected, and the end product is there. And here you see this dull little plant. This is the wild cabbage. Now, we can assume that everything is solved and we understand how evolution works, and we can explain why the traits are there, under which kind of selection they have evolved, and so forth. So I could stop here. However, there are a few exceptions. Unfortunately, one of these exceptions is very widespread. I usually call it the best guarded secret of biology. We do not understand why most plants and animals reproduce sexually. Especially, we cannot explain why the male sex exists. We would have no problem if the world consisted of parthenogenetically reproducing females, because this is much more efficient, and it's easy to show why it is so. Let's assume there are two females, a sexual one and an asexual one, and both produce two offspring each. The sexual one produces a daughter and a son, and each of them inherits only half the mother's genome because of meiosis. The asexual female produces two identical copies of herself. That means two daughters which are 100% identical genetically. Now let's look at the next generation. Only the daughters, but not the son, produces two offspring each. So you see here that in the generation of grandchildren, the asexual female is already twice as successful as the sexual one. And this is because males do neither give birth nor lay eggs, so they do not contribute materially to the reproductive success of their mother. Sons seem to be a luxury that should have been removed by natural selection a long time ago. And if we look at further generations, it seems hopeless for sexual reproduction. However, I told you that what we see now, which has evolved, has to be assumed to be the best solution. So why is the best solution so inefficient? Let's have a look what a sexual female does when she decides to reproduce. So first of all, she throws away half of her genes during meiosis because oocytes have only one haploid set of genes. And then she spends a lot of time and effort, which we call display behavior and partner choice, to find somebody else who has done the same, namely has thrown away half of his genes to produce gametes, and she fills up from him what she has thrown away before. Ridiculous. If an engineer would design reproduction this way, you would fire him. Now the only hope is that she selects that individual from whom she buys the new half of genes in a way that the offspring she is producing now are qualitatively at least twice as good as if she would have reproduced asexually. So she has to compensate this efficiency disadvantage compared to asexual reproduction by a quality advantage. Now, in order to work, three conditions have to be fulfilled. First of all, the environment has to change dramatically with no end, so that it is worth throwing away each generation a new half of her genes and buy a new set and mix. If this change of environment would stop, then you would just keep what you found out last generation was the best solution. So then we would switch to asexual reproduction. So the environment has to change continuously and dramatically. Second condition, the genes with which we respond to that environmental change have to be polymorphic. We are usually not polymorphic. Our genome is 99% identical to that of a chimpanzee, so that 1% is the difference. That means any two humans are very similar genetically. We are not very polymorphic. And third, the reason why we have to be polymorphic is if you throw away something and you want something different and you want to choose, that means that there must be potential partners which have different sets of genes so that you can go for the optimal complement. And third, as a butterfly or fish or even human being, you should be able to recognize these genes, version of genes, in potential partners without using next generation sequencing and so on. Well, this sounds like science fiction, but it works. So what's the environmental change? The great evolutionary theorist from Oxford, Bill Hamilton, had the idea. He said, it can't be any physical, it's too slow, climate change, it's too slow. And he suggested it is infectious diseases. They come in large numbers upon us and change all the time. And he said, it's a miracle that we are still alive. So there is strong changing selection. Now, what about immunogenes? Are they polymorphic? At least in vertebrates, there are the genes of the major histocompatibility complex, MHC genes, which are part of the adaptive immune system. And these genes are the most polymorphic genes that have ever been found in vertebrates. There are not just 5 versions as for blood groups or eye color or whatever. There are more than 1,000 alleles in each vertebrate population that had been looked at. And in human populations, we have up to 3,000. And each of you got between 6 and 8 of these alleles. And everybody has probably a somewhat different collection of 8 alleles of these 3,000. So we differ. And now the next condition, are we able to recognize in potential partners which versions of MHC alleles they carry? Now in the late 70s, the American immunogeneticist Kunio Yamazaki found just by chance that a mouse prefers the partner which offers different MHC alleles that are different from the one she's got herself. And he found exactly the same result when he offered only the urine of these 2 partners. That means mouse can smell MHC alleles in potential partners. And the fact that she preferred different shows that she knows what she's got herself. Well, this is marvelous. Now the MHC system is highly conserved in all vertebrates, starting with the sharks. But it's very similar in mice and fish and birds and reptiles and in humans. And we can study it with any vertebrate. In those days, I had the pleasure to the permanent position at the University of Bern. Nobody understood actually why I left, but at Max Planck you can really do only research. And in our seminar, we discussed Yamazaki's results and came to the conclusion, if this is the mechanism that maintains sexual reproduction, in vertebrates at least, then it should be the same in humans. And it's very easy to study this in humans because you can buy the MHC typing. It's called HLA, human leukocyte antigen in humans, but it's the same as MHC, and I continue to call it MHC. You can buy this typing from each hospital that does transplantations. That's, by the way, the reason why transplantations are so difficult to find a fitting donor for a receiver, because we are all different, our immunogenes. And I had a very highly organized PhD student, Klaus Wedekind, who said he can organize such a test. And he found 144 volunteer students who were sent to the hospital, their blood taken, analyzed, and then he provided the males with a set of 2 T-shirts and rules how to behave for 2 days at least, smell-free, not smoking, no alcohol, no garlic, no spices, and other things. And then they had to sleep alone wearing such a T-shirt in their bed for 2 nights, 2 T-shirts, and then they were asked to send the T-shirts back in a plastic bag to the university. And he asked each woman to come to the institute in the middle of her cycle, because it's known that the sensitivity for smell is greatest then. And each woman was presented with 6 of those T-shirts. 3 had been worn by MHC similar males, as similar as possible, and 3 by MHC different males. And for each T-shirt, she had to mark something between very unpleasant to be until very pleasant, somewhere in between. And to make a long story short, it was exactly the same result as Yamazaki has found in mice. Here you see the pleasantness of male orders. By the way, I forgot to say, each male was used twice, therefore 2 T-shirts, once as an MHC similar male for 1 female, and the second time as an MHC dissimilar male for another female, just to avoid that an absolute stinker could bias the results. So this would cancel out. And now you see that's the pleasantness ranked, and the stippled line is neutral, and everything above is attractive, and everything below is unpleasant. And you see that the T-shirts of MHC dissimilar males were ranked pleasant by these females, and the ones worn by similar males were unpleasant, as in mice. And you see here, this is true only for women who did not take oral contraceptives. And for those who took oral contraceptive, the result was the other way around. So they found MHC similar males more attractive. And again, this is exactly the same as Yamazaki found in his mice. Not that mice are taking contraceptives, but you know that better than me. In those days, the pill mimicked pregnancy. And when you're pregnant, you're not searching for partners, you're searching for social partners that help you raise the young. And pregnant mice preferred MHC similar social partners, which means relatives. Now he asked for each T-shirt whether it reminds the woman of her actual mate or ex-mate, and here you see for those which did not take the pill that those T-shirts worn by MHC dissimilar males reminded them significantly more often of their actual mate, for which he concluded that mate choice under more natural conditions with garlic and all this have worked in the same way. Now we had a problem. At least I saw the problem. Perfumes. So actually, at the time when this wonderful mechanism starts working at puberty, we start using perfumes with which we change our personal order. And I thought, now I of course studied the literature and found out that perfume use was the usual habit in all societies around the world, at least since the start of written history, which is since at least 5,000 years. And in all societies, about the same 100 strange ingredients have been used for producing perfumes. There is no fashion in perfume use. So Chanel No. 5 is approaching the age of 100. There are other bestsellers that are even older. For example, Shalimar and Mitsuko are over 100 years old, and all the rest of the bestsellers are also quite established. And it is known that people have a huge problem to find a perfume that they like on themselves. You might have made the same experience. You like many perfumes on other people, but for yourself you are highly selective. So for me, that smells as if MHC alleles, our MHC alleles dictate which perfume ingredients we have to use to amplify our personal perfume. And these individual differences in preference for personal perfume are well established. After people found their perfume, they usually stick to it for 10 years or even longer. And many experiments have been done to correlate these individual differences with personal traits such as temperament and skin type, hair color, and whatsoever. Nothing explained it. So I thought, because we are using perfumes for such a long time, they have become part of our biology. And I convinced Klaus that we do another test with his volunteers. By the way, why do we use perfumes? So if you think of Louis XIV, to cover bad smell, that's not the main reason. If you believe in the PR campaigns of the perfume industry and in the Bible, if you read Solomon's song, Please Do, you will be astonished. So actually we use it to increase our sexual attractivity. And one of the oldest recipes has been written down in a book of Moses, and it's really, you can produce the perfume with this recipe, and there are 2 versions of it still on the market which are written down there. So it's no fashion at all. Everything is there for a long time. We bought 36 natural perfume ingredients, no synthesized version, natural, very expensive ingredients, and then had 36 glass tubes, each containing a smelling strip, and to each strip we put 2 drops of one of these ingredients, which had been diluted in the way as if they would be used in perfumes. And then each of the 144 students got this package and was asked, for each of these substances, would you like to have this substance in a perfume or aftershave you would like to wear on yourself? Would you like to smell like that for each of them? We thought it's quickly done, 2 days. No, it was not. So 144 times 38, this is something. We spent a whole week, here you see Klaus Wedekind preparing the strips. And then we got the results, and he did not believe in this idea. He analyzed them and was rather surprised. For the most frequent HLA alleles in humans, 4, we found highly significant correlations with the spectrum of preferences people with this allele or that allele had. Highly significant. So it seems as if actually our MHC profile explains our individual differences in perfume use. And it seems that they dictate what we find attractive on us. Please try to use one which you wouldn't use on yourself. Wear it for a day. You can't stand it. You would vomit. So our physiology tells us what is best for us. And the idea is that these perfumes amplify the natural order. And this is a painting 150 years old. It shows you that people who prefer different ingredients, left is tuberose, right is jasmine, find each other attractive. And here, this is very interesting. It shows that there is a tradition of perfume ingredients from the mother to the daughter. And I found a description of that in the Perfume Museum in Paris. And it was said that in the Middle Ages, perfumes were not very stable, so people had their preferred 6 to 8 different ingredients, and whenever they needed the perfume, they mixed them and used them, and kept these ingredients for the lifetime. And then, believe me or not, I could read that the mother hands over her tubes to the daughter, and the daughter usually exchanges half of them for other ingredients. Of course, she has inherited only half of her MHC alleles from her mother and the other half from the father. So this makes absolute sense. There are several take-home messages. One, if you enjoyed reading Süskens The Perfume, the final end can't be true. This is absolutely mistaken. There can't be a perfume which renders everybody most attractive to everybody else. It depends on your MHC alleles and the MHC alleles of the receiver, which is attractive and which is not. And another take-home message, never donate a perfume to someone else that you have chosen yourself. It would end in a disaster. The person will never find this usable. Now, let's become more serious. What do MHC molecules do within our cells? If you suffer from a new infection, then your cells contain foreign proteins. And these proteins are degraded by proteasomes into small pieces, usually 9 amino acids long, peptides. And these pieces swim in your cytoplasm, but the rest of your immune system is outside the cell, your T lymphocytes. Now it needs something to grab these peptides and transport them through the cell membrane and show them to T lymphocytes. Otherwise, they would never find out what the infection consists of that you suffer from. Now this is done by MHC molecules. So you have between 6 to 8 alleles, and each allele programs for one specific type of MHC molecule. They are very similar, but they differ in the form of their binding groove with which they can bind to these peptides. And only if the amino acids at certain positions, and the most important ones are position number 2 and number 9, fit exactly into the binding groove of one of your MHC molecules, the molecule can bind that peptide and transport it through the cell membrane and show it to T lymphocytes. Now, T lymphocytes can distinguish between foreign and own. Your MHC molecules not. They grab every peptide of which the anchor residues, these amino acids, fit into the binding pocket. So if you're not infected, they show your own peptides. And now T lymphocytes go along and say self, 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 foreign. And after they have detected one foreign one, then the cascade, you know, from the vaccination response is induced, and after 6 to 8 days, you have built up an armada of highly specific cells that detect infected cells and destroy them. However, if the situation is like this, then you have a problem. Then you have to, this would not fit, and then you have to rely on your innate immune system, which is much more expensive and not so efficient. So it's good to have as many different MHC alleles as possible, obviously. Then your MHC molecules can detect and show a broad spectrum of infectious diseases to your T lymphocytes. So it seems to be a good idea that Yamazaki's mice and Klaus' Bernese students preferred partners that had different MHC alleles from themselves to increase the heterozygosity for their offspring. Now, is this enough to produce this double quality advantage through mate choice? There would have been a very simple way to test for it. We should have asked each of the women in the Bern experiment to produce one child with one of the 3 men who were MHC dissimilar, and one child was one of 3 men which were MHC similar to that woman. And then we would have to follow these children through their lifetime, at least the first 45 years, and list all infectious diseases and how much they had to be treated, and of course count all the offspring that these people produced themselves. Now, this is even in Switzerland not possible. The MHC typing was quite expensive. It was 1,000 Swiss francs per person, but we were in Switzerland, so that was no problem. But doing this experiment with humans is unacceptable for many reasons. But as I told you, the MHC is the same in all vertebrates, so we can do the same experiment, for example, with fish. And after I moved to Plön. Plön is surrounded by 400 lakes, and each has a stickleback population by its own and accompanying parasites. And these fish are famous for their conspicuous display behavior. Here you see a gravid female, and she is surrounded by a male in bright breeding coloration doing his zigzag dance. For example, Nico Tinbergen got the Nobel Prize for describing and studying this behavior in sticklebacks and other things. So we studied these fish. We had to develop the MHC typing and found that they have about 6 copies of these MHC genes that are responsible for detecting macroparasites. That means if you have 6 loci, then you have 6 alleles from your father and 6 from your mother. And if these fish had chosen partners as mice and students, they would be different at all loci, and we would expect 12 different copies per fish. We caught fish from the lake and counted alleles, different alleles in these fish, and this is the expectation that the class with 12 alleles should be the most frequent. And what we found is this. Disappointing, isn't it? So we found fish with an intermediate number of 6 alleles to be most frequent. This does not look like maximizing MHC heterozygosity. This looks like an optimum. And actually an optimum is predicted. We had not, and others had not paid enough attention to a PNAS publication by Martin Nowak et al. From 1992, where he predicted this. If there are too many different MHC alleles per individual, only few T-cell lines survive because of negative selection of T-cell lines in the thymus. And as I told you, MHC molecules show your own peptides, and each of your T-cell clones is tested in the thymus whether it responds too strongly to such a combination of MHC molecule with self-peptide. And if this is the case, then the whole clone is eliminated. And with your 6 to 8 different alleles, already 95% of your original T-cell repertoire has been removed, and all the attacks have been done by the surviving 5%. You can easily imagine if this number of different MHC molecules further increases, then there is no army left in the end to attack infected cells. So there is this optimum. Now we should have counted all the parasites in and on these fish from the lake, and we would predict that the ones of the most frequent assumed optimal type are least infected. We did an experimental test for that, so we raised clutches of offspring from wild-caught fish with a broad spectrum of alleles per fish, and then exposed them, each young fish, twice individually to the same dosage of the 3 most important parasites of these sticklebacks from the lake of their parents. So we were sure that each fish had eaten the same amount of intermediate hosts carrying these parasites. And we did this twice after a break of 6 weeks to allow for the vaccination effect. And then after 2 further months, we dissected all the fish and counted the parasites. And this is what we found. And it looks really as predicted. So fish of the most frequent intermediate type, 6 alleles, had the lowest number of parasites, species and parasites per species. And the ones to the left had more, and the ones to the right had more. The ones to the left because they had too few different MHC molecules, and the ones to the right, they had enough MHC molecules, but their T-cell repertoire had been deselected. Now we did the same thing also with fish, not infected in the lab, but released when they were adults into these cages in the lake, and these fish were exposed to all the parasites in the lake. And then during their reproductive lifetime, which is 1 summer, we collected every week all the clutches from all nests, and because we knew the microsets of the parents, we could determine for each egg who was the father and who was the mother and make for each fish a list. And at the end of the stickleback's lifetime, we could spot the most reproductive fish, and these were fish close to the optimum. So it's possible that this double quality advantage is achieved through mate choice for complementary MHC alleles, at least in sticklebacks. Now we want, of course, know whether this distribution in the lake has been produced by mate choice or whether it is just the product of parasite selection, the better ones surviving until we caught them. And being vertebrates, and because the MHC system is so conserved in all vertebrates, we assumed if mice and humans can smell MHC alleles in potential partners, fish should be able to do that as well. If we had asked for money from the German Science Foundation, they might have found that ridiculous because, of course, everybody knows that fish, and especially these fish, are oriented only visually. They are so bright and have this dance and everything. Why smell? Because this is conserved, and we think they have to use the same mechanism as other vertebrates. Now, this is a T-shirt test for sticklebacks. This is a long flow channel with a constant current, and here is a partition until this kind of cage starts, and we could put a gravid female into this cage and observe her behavior via this video camera, and then we could add to each of the two halves water that we had taken from above the nest of a displaying male and add it constantly to the current for 5 minutes. And then after 5 minutes, the sides are swapped to make up for side preferences and so on. Now I show you a short video showing just the cage from above. Here is the fish, and here will be the water of one fish appearing, and here of the other male, about 20 seconds after this red lamp had been alighted. Now we start. Now the lamp is on, 20 seconds from now, nothing to be smelled now. The female is relaxed, explores, is bored, wants to leave. This is downstream. So about now, the order of the two males should have arrived in the middle of the cage and flowed slowly to the end. Now let's have a look whether she recognizes them. And from now on, there is nothing else than trying to get upstream. This is the case in all females that we tested. Obviously, she smelled something because we have only smell presented of the males. But now take into account she does not know what is offered above. Only after she has tried this other side, she would be able to compare. But it's already okay what she smells. She wants to get there. She likes it as well. Seems a bit more enthusiastic. We had females who got stuck in these strings. And now something happens what never appears on stickleback life. She can't get there. You can imagine this question mark in her brain. How do I get there? Try it again. Can't be true. But let's wait until she tries the lower male again and whether she still likes it. Maybe she likes everything. But she will go there, I know the video. No longer attractive. So this is the one she likes, and I can assure you after we had evaluated the results also for this female, she strongly preferred the upper male. Now we did about 80 such experiments with each time a new triplet of two males and a female, MHC type, we could determine with which of the two she would come closer to the optimum for her offspring. I developed an algorithm with which I could calculate whether there is an optimal number that all these 80 females taken together try to achieve. And the result is there is an optimal number of different MHC alleles that females try to achieve through mate choice. This is highly significant. It's complicated, so I skip this procedure. And then you can have a look where it is. And this optimum is close to 11 alleles if you mix all alleles of the male and the female. So we can conclude female mate choice, at least in sticklebacks, optimizes by allele counting with self-reference to achieve female plus male together approximately 11 alleles for the offspring. And because each parent provides haploid gametes, you have to divide this number by 2, and that means that the offspring inherit on average 5.5 alleles. And you might remember this is very close to the most frequent type in the population and very close to the experimental optimum that we have demonstrated. By the way, this optimum had been shown in a number of species until now, so it seems to be everywhere, and we guess it will exist also in humans, what we studied just now. And now the $100,000 question, which is the natural molecule which transmits by smell, by odor, the information about its bearer's MHC composition. The mice people, of course, analyzed a lot, so they took the urine and analyzed all the peaks that they found, usually 1,000. Well, this is finding the needle in the haystack. I teamed up with Thomas Böhm, he's director at the Max Planck Institute for Immunobiology at Freiburg, and we thought we should have a look at the physiology and where is a molecule which could do it. And this molecule has to be as polymorphic as the MHC itself. And we came up with the hypothesis the peptides might be the signal. And you know, there is a one-to-one lock-to-key relationship between the binding specificity of the molecule and the nature of the anchor residue, amino acids, at these positions. So if you measure all the MHC ligand peptides that leave a male, and they can leave it only after being transported by an MHC molecule, and you determine the anchor residues, you could in principle tell which alleles that male must have possessed. And if it's possible in principle, we can assume that females and in other species males as well have evolved such a screening mechanism. So we tested that idea. Here you see my colleague Dr. Sian Griffiths, which did part of the following experiments. We were sent pairs of tubes with one containing a mixture of 4 different synthesized MHC ligand peptides in solvent and the other only solvent. And each pair of tubes was coded in Freiburg, individually coded, and the code was retained until we had analyzed the results. And now we used only the water of one male for each female. And on one side of the flow channel we added 4 peptides, but we were blind, but we knew one would contain peptides and the other solvent, but did not know which is which. If a male has too few alleles for that female so that she can reach the optimum for the offspring, and if peptides mimic additional MHC alleles and are counted as additional MHC alleles, then she should prefer the site with the peptides, because then that male mimics a male that is closer to the optimum complement that she looks for. However, if the male already offers enough alleles that she can reach the optimum, or is even above that, then the peptide site would mimic a superoptimal male, and she should avoid that. And in this case, she should go for the peptide-free site. So it depends just on the MHC composition of the pair, whether adding peptides is positive or negative. This is the prediction. It's a control in itself. And here you see the preference of the females for the peptide site. We had altogether 10 minutes, this is 600 seconds, and you see here how many of these 600 seconds she spent on the peptide side. 300 is neutral, and here we have pairs with combined numbers of MHC alleles. So this is female plus male. For the optimum, you have to divide that by 2. And here you see pairs below the optimum. And in these cases, the females preferred the peptide site. And pairs which were already at the optimum or above it, females avoided the peptide site. This is quite significant, and it suggests that these added MHC ligand peptide are treated as if they would signal additional MHC alleles of that male, and we guess that it also suggests that these peptides are also the natural signal. Now, Thomas Böhm had another collaboration, our sister publication. He looked for the receptor site in the vomeronasal organ of mice, and they found a population of highly specific olfactory receptors, and each responded to only one MHC ligand peptide. And here you see that. So this is the sequence of amino acids simply. It's 9 amino acids. And here you see the response of the receptor to this one, and here the response of that receptor to this one. And now the test is he exchanged the amino acids at the anchor, which served at anchor residues, for alanine. Alanine never functions as an anchor. And you see, if the rest of the peptide is kept constant, but the anchors are exchanged for alanine, the receptor is silent. And now you can do the reverse experiment. You keep the original anchors and exchange all the rest. And the expectation is then the receptor does not distinguish between wild type and mutated. And this is shown here for one of those. So this shows that the amino acids at anchor position that defined the specificity of binding MHC molecules are decisive for these olfactory receptors to respond. This is exactly what we would expect if on the receptor side. We did an experiment with sticklebacks where we used these mutated peptides. So this is the mix that we have used before, wild-type peptides synthesized, and here the same thing, but all the anchors mutated to alanine. And if these are not recognized, then they should work such as solvent, water, and we should find the same result as before. If they are recognized as wild-type peptides, then we should find always 50-50. And as you can see here, it's not the same figure, it's a different one, but the results are very, very similar. So also on sticklebacks, anchor residues and MHC ligand peptides are decisive for recognition. Now, conclusion, the immune system peptides act as pheromones, both in mice and in sticklebacks, and therefore probably in all other vertebrates, including humans. We are studying this at the moment. They are probably the natural chemosignals, perfumes, that transmit olfactory information to choosing mates about an individual's MHC immunogenetics. Now we come back to perfumes. This is a very interesting book, The Scented Ape, It's Us, by Michael Stoddart, and there's everything from neurophysiology to perfume use. And he wondered in the perfume chapter, there are thousands of well-smelling flowers, and none of them is used in perfumes. Why do we use, since 5,000 years, only about 100 natural ingredients of rather strange origin? What is special in these ingredients? Here you see Chanel number 5, and the components are not a secret. You can find them. And here is the base note, and here is, for example, amber. I don't know whether you know what amber is. Amber is what sperm whales spit out as undigestible of their prey. And fishermen caught these sticky things and got the weight in gold after that. And this is one of the most famous ingredients. And all the others are also quite irritating. Even Yasmin as absolute has a fecal odor, so quite strange. Now, the answer might be, what is special in these ingredients? Maybe one of the components is a mimic of an MHC ligand peptide. Not chemically, but it elicits the same receptors, such as sugar and the stuff that we take instead to save calories. I have to thank all my collaborators and you for your attention, and especially that you came to this lecture after this marvelous dinner last night. Thank you very much.